Achondroplasia is a form of short-limbed dwarfism. Similar physical traits shared by achondroplasia patients include a larger head, shorter limbs, and an average-sized trunk. In this video, we will discuss how a gene mutation causes the short limbs in the achondroplasia patients. So the mutation in genes, it turns out, codes for a protein that's called fibroblast growth factor receptor 3, or FGFR3. FGFR3 belongs to a family of receptors that can be found on the cells of various tissues of the body. Those cells all express different genes that are in charge of producing different receptors, which means that those cells all have different receptors on them. These receptors are specific for different growth factors. When the body wants to regulate the development of a specific tissue, it sends out the growth factor, which will be picked up by the receptors of the cells on the specific tissue and would cause the occurrence of signal transduction pathways. So our protein FGFR3 is mainly found in chondrocytes, which are cells that are embedded on the cartilage tissue. Cartilage tissue is a tissue that goes through ossification to form bones. In ossification, the cartilage growth plate develops into an actual bone when the chondrocyte cells proliferate and differentiate. On a chondrocyte cell, the chondrocyte cells express the FGFR3 gene. That means on the cell membranes of the chondrocyte cells, we would find our protein receptor. And the protein receptors are specific for the growth factor FGF ligand. And the protein, our FGFR3 is actually, it turns out that it's actually inhibitory to cell growth and cell proliferation in cartilage. This happens when the ligand comes to activate our protein receptor. It also activates the occurrence of several different signal transduction pathways. One of the pathways involves the activation of the molecule STAT1. The activation of STAT1 would enhance the expression of the molecule P21. P21 is an inhibitor of CDK. When P21 binds to CDK, it gives a conformational change to the CDK active site for cyclin, causing cyclin to be unable to bind with CDK. Normally, the complex of cyclin and CDK would allow the binding of another complex of RB with E2F to bind to the complex, which causes the dissociation of the two molecules, leading to the release of E2F. E2F is a growth factor for various enzymes that are in charge of DNA replication, such as DNA polymerase. When P21 binds to CDK, it prevents the complex between cyclin and CDK from forming, which in turn prevents the release of E2F. When the cell is unable to replicate its DNA, it is also unable to enter mitosis, because mitosis requires the cell's DNA to be replicated first. Therefore, it can be said that our protein FGFR3 is a negative regulator of cell division and cell proliferation.
So in a patient unaffected by achondroplasia, as the 1138 base pair, there's a guanine, and within that guanine is in a codon, which codes for a glycine amino acid. However, in the patients that were affected by achondroplasia, in the 1138 base pair, there was either a C or an A instead of a G. And these two codons both code for an arginine amino acid instead of a glycine amino acid. So these two amino acids change the primary structure of a protein, and therefore, because arginine and glycine don't have similar properties, it changes the overall shape of it or the tertiary structure of the protein. So this single change in a nucleotide is called a single nucleotide polymorphism, which is an SNP. So basically only one nucleotide is mutated. In this case, it is a missense mutation because it changes the amino acid, and the change in the amino acid affects the formation of the protein. As you can see here, it changes the tertiary structure. So the change in the protein mutates the FGFR3, and it is no longer ligand dependent, it's ligand independent now. And so it doesn't need the FGF ligand to bind to it to start the signal transduction pathway, and it on its own starts to create STAT1, and STAT1 enhances the expression of P21. And so more and more P21 is created, and P21 is the allosteric inhibitor for CDK. So therefore, when it binds to CDK, cyclin can no longer bind to CDK, and therefore RB and E2F, the cyclin on the CDK is no longer able to release the E2F from the RB. And the E2F is a growth factor for the enzymes that are in charge of DNA replication. So when the E2F is enabled to start the DNA replication, the cell isn't able to go through mitosis, and therefore cell growth or cell division will stop, and bones won't be able to form. So overall, the mutated FGFR creates more STAT1, which creates even more P21, and the combination of both of those creates over-inhibition, and therefore bones aren't formed to their full potential.